We're back. We're in Chapter 9, The Methods of Microbial Control, Part 2. We're still talking about the physical methods, and we're picking up with pasteurization, another moist heat application of physical control. We've talked about pasteurization before, but we're going to talk a bit about the details. Please become very familiar with these details. Pasteurization is used for a lot of fluids, like milk, which probably is what you associate with the term. However, other products are pasteurized as well. It's important to realize that pasteurization is not sterilization. Normally, heat-tolerant organisms will survive, but the pathogens should be killed. When we're considering milk, all 4 common types of pasteurization might be available. These methods are used for other products as well. The batch method is the old school method, perhaps still used by mom and pop type operations. For this method, you take a big batch of your product, like in a large vat, and you heat up the product to 63 degrees Celsius. You have to hold it there for 30 minutes to pasteurize the product. The disadvantage to this is that it can alter the flavor of the product, like milk. The milk can take on some cooked types of flavors, which are perhaps not desirable for your product. It is the lowest tech and most basic approach to pasteurization. These next 3 methods try to overcome the cooked flavor drawback by applying the heat step in much more quickly. But to speed up the heat step requires higher temperatures. Flash pasteurization will heat up the product to 72 degrees Celsius for 15 seconds. The way this is done is using heat exchangers. So let me try to draw a little diagram on the slide for you to demonstrate how heat exchangers work. A heat exchanger uses metal plates that are held at different temperatures. Tubes of the fluid run right next to these plates. The first plate would be the hot temperature that you wanted to reach very quickly. Then there would be a colder temperature on the second plate. You would have products running through the tubes that are laid across the plate, and it would go back and forth across that particular plate, so the fluid inside the tube would reach the temperature very quickly. The rate of flow is controlled so that it's held at the temperature for the amount of time needed, and then the fluid passes over the cooled plate. The tube with fluid run back and forth so the fluid chills back down quickly. The fluid running over these plates heats up very quickly and cools down very quickly with this method. You can use all of the different temperatures that are listed in the next 3 methods. You could do 72 degrees for 15 seconds, which is the flash pasteurization. Ultra high temperature is 135 degrees Celsius for 1 to 5 seconds. If you wanted the product sterilized, then you could raise it even further. If you notice, in the grocery store, these boxed milks that are left out at room temperature for very long periods of time, they obviously have to be sterile. Otherwise, you would have spoilage in those products because it's not refrigerated. So you're talking about a very high temperature of at least 135 degrees Celsius for a few seconds to achieve a type of pasteurization that essentially is sterility. Next, we're moving on to dry heat. Hopefully these instruments look somewhat familiar. Over on the left, you have an incinerator that you use to sterilize your loop. When using it, you avoid generating aerosols because anything that pops from the loop is captured inside the chamber and burned. A Bunsen burner flame might potentially cause something to aerosolize with the updraft generated by the flame if it's not killed immediately. Dry heat is used for materials that can't be sterilized with moist heat. It does the same kinds of damage, denatures proteins, ruins membranes and nucleic acids, but it's going to require a higher temperature for longer. Obviously, the 2 items shown here burn everything. They incinerate something, so you can't use it for some items. If you want to sterilize loop, it works perfectly, but you're not going to be able to sterilize something like your lab coat with this method because you'd burn it all up. On the opposite spectrum is cold. If we're going to chill something down, then when we cool the temperature down, we are decreasing microbial metabolism and slowing down all of those chemical reactions, including all of the enzymatic reactions within the cell. If you place microbes in the refrigerator, for the most part, you're going to halt the growth of most pathogens. I've mentioned the exception, the term psychotrophs. The psychotrophs are the microbes that have an optimal temperature in the mesophilic range, so the range that we exist in, but those can grow quite well in the refrigerator. This can be a problem, like the pathogen Listeria monocytogenes, that can be found in the environment. It could contaminate a food product, and then you put that food product in the refrigerator, and it doesn't really slow down the growth of the pathogen much. The organism can increase to high numbers in the refrigerator and then make somebody sick after eating the food. Slow freezing will kill more organisms because you get more ice crystals damaging cell membranes compared to a rapid freeze. Organisms vary in their susceptibility, but freezing will not kill organisms overall. You probably have a sense of this already. Think about buying a chub of something like raw hamburger and sticking it in the freezer, and then thawing it out. You know it's not sterile. You wouldn't dare eat it or feed it to somebody like that without cooking it, right? In some sense, you already knew and you've already had experience with the fact that organisms will survive that freezing process. Freezing is not a way to sterilize food. The best way to preserve microorganisms is actually by desiccating them during freezing process. Freeze dry them. If you're actually trying to preserve a culture, freeze drying them, which is properly termed lyophilization, that's going to preserve organisms for decades. That's how different companies and different scientific organizations keep specific strains of interest preserved for future study. They lyophilize them. You can purchase organisms as powders from lots of commercial sources. You just add them to broth and then they begin to grow again. By freeze drying them, you're preventing any kind of damaging ice crystals. So while you might be able to keep organisms in a deep freeze, like -80 for a few years, you could keep them lyophilized for decades, and they would still stay viable. If you desiccate a product, you obviously prohibit growth because life needs water. Another method of sterilization involves physical methods like filtration. Here you see a filtration apparatus. Below, you would have to have sterile capture containers. Everything below the filter must be sterile. The filter would be placed at the bottom of the chamber where you add the media. Then you filter media through. The fluid at the bottom would now be sterile. The electron micrograph is showing you a filter with some pores in it, and it has trapped some spirochetes on the surface. In the earlier chapter, I showed another filter with trapped bacteria. That was to illustrate how to capture bacteria in a fluid to count them, but the filter works the same. Trap bacteria while allowing fluid to move through. This image allows you to see the size of the pores in these different membranes. You might wonder why you would filter something instead of autoclaving it. Sometimes heat will denature something important in your media. Perhaps your cells need some sort of growth hormone or organic molecule, something like that added to your media. You can't sterilize those types of molecules or you'll ruin it, denature it. You would have to do something like filtration in order to ensure sterility before you use that particular media. This table from your book is showing you different types of membranes that can be purchased from a commercial operation. The pore sizes vary. As you might imagine, the smaller the pore size, the more expensive the filters become. In general, what we use in the microbiology lab is going to be in this range right here, about 0.45 and 0.22 micrometer pore sizes. That size opening is going to trap bacteria and not allow them to come through the filter. So the media will be sterile after passing through. Of note, viruses are much smaller than bacteria and they're not trapped by the filtration. Viruses, if present in your solution, will also be found in the filtered media. This apparatus, shown here, is a biosafety hood. We have one of these in the back prep area of the microlab. This piece of equipment is really important for protecting the individual using the hood from anything inside the hood, but it also would protect the work you're doing inside the hood from any external contamination as well. Let's examine the key features. First is the HEPA filter. The HEPA filter, HEPA stands for High Efficiency Particulate Air Filter. HEPA filter is going to capture all of the different particles that could be in the air, including viruses. Next, look at the airflow. If you look at the arrows, you might notice that the airflow is all contained within the safety hood. You will not have any air blowing out, which is to prevent contamination of the individual if you're working properly in the hood. The person outside is protected from contamination by basically a curtain of air that's blowing downward. Inside, there's a flow of air circulating and going through the HEPA filter. This biosafety hood is a very important, albeit a somewhat expensive piece of equipment to work with microorganisms. Another type of physical control is the use of osmotic pressure. You can see here a food, a jam, that has high sugar content, but you can imagine a high salt content as well. The method is important, but it's not going to be fail-safe. As you can see in the image, there's a mold growing in the jam. It's true, the fungi in general do tend to be more resistant to the osmotic stressors than, say, bacteria are. Nevertheless, using high salt or sugar to impose osmotic stress is a common method of preserving products that's been used for a very, very long time. The next physical method is radiation. This term is really scary to people that don't understand what it is. Radiation is a form of energy. When you microwave your food, you're subjecting it to a form of radiation, but the radiation that I'm referring to here is ionizing radiation. That means it's high energy, higher energy obviously than microwaves. These radiation processes involve electron beams, gamma rays, or X-rays. These processes are termed ionizing because the high energy waves knock electrons out of orbit, creating ions. These ions react. The energy might also break bonds in molecules, such as DNA or proteins. This damage will kill cells. However, the food itself is not radioactive after the process, just like your microwave dinner is not releasing microwaves. Gamma irradiation is a cold process, unlike microwaving, so it can be used to preserve food by killing spoilage organisms or insect pests or even pathogens. If products are treated via one of these ionizing radiation methods, then it needs to have this Radura symbol on the product. This green symbol, it sort of looks like a flower. That's called a Radura, and that indicates that a product has been irradiated for safety purposes. There has been much research done which shows that these processes obviously are safe for the consumer and effective in reducing microbial load. You can see in this image of irradiated and non-irradiated strawberries, the strawberries would last a lot longer after treatment compared to one set that has not been treated. While the process has not been shown to generate anything harmful, it does potentially have the ability to change some flavor profiles. Some foods can't be irradiated. Nuts, for example, take on a flavor like residuity that kind of tastes like it's old and people don't like them. Thus, the process can't be used on all foods, but it's not a safety issue. Tons of studies have happened to show that it's not going to cause a safety problem in the food, which makes sense from a scientific standpoint. This is just another form of transferring energy like cooking, microwaving, etc. Despite the hype that's out there, there's research to show that it's really not a safety problem and it's perfectly safe to use it under the appropriate conditions. But regulations state irradiated products need to be designated with the Radura. There is an exception, which leads me to make the following statement. You all have had some irradiated product in your diet already. If at any time in your life you have eaten at a fast food restaurant, you've had some. That's because the irradiated product is in very small amounts in a food matrix. It doesn't have to be labeled then. Commercial spices are almost always irradiated to avoid bringing contaminants into the food. Spices are typically pretty high in microbial numbers. So spices, and that includes things like pepper, are irradiated for commercial purposes. If you're a commercial entity like a fast food restaurant, you're adding a lot of spice to a big batch of product like taco meat, and you want to avoid introducing a large number of microorganisms. So commercial entities tend to use irradiated spices to minimize that danger. Spices are a very small proportion of the overall product, so they don't have to indicate that the spices have been irradiated. Now that you know about this symbol, you might want to take a look when you're in the grocery store and see if you can find anything that has it. I guarantee you will not find it at Trader Joe's, Whole Foods, and similar stores who cater to a certain type of customer. Those customers would reject the process. But I know that other stores, there are a few fruit products that come from other areas of the world that need to be treated this way so that they don't bring any insect pests that could damage the agriculture in California. Guavas are one. If you find guavas, they are likely in a bag and they have the Radura symbol on the bag. There are also some beef products, especially from the Midwest, that are treated this way. In the Midwest, they have suffered some very large-scale recalls that have devastated their industry, so they're more accepting of beef that's irradiated. They realize the protective nature of the process, that the treated products are free from pathogens like E. coli O157H7, a strain that causes serious and sometimes deadly infections. Midwesterners are thus more accepting of irradiation because they recognize the value of the treatment compared to consumers here in California. In California, many consumers get very vocal about what they perceive as unnatural means of treating something. Non-ionizing radiation uses lower energy wavelengths. The wavelengths are longer. Think UV light. That's the most common non-ionizing radiation in use. You get exposed to it every time you go out in the sun. When you're in the sun, you're exposing your skin to UV light if you don't have on sunscreen. It's not penetrating into deep tissues, but it does penetrate into the skin. What's happening is that the wavelengths of energy create improper bonding in the nucleic acid strands. This image is illustrating a pyrimidine dimer. The strand is bonding improperly. Rather than having proper hydrogen bonding between the nitrogenous bases, you get adjacent nitrogenous bases forming a covalent bond. This occurs when UV light hits the strands at adjacent pyrimidine bases, the T's and the C's, but the bonds like to form most often at the T's. If these improper bonds are not repaired, it will lead to mutations. The dimers are what leads to things like skin cancer. If enough of these are formed in the skin cells, your skin cells are not able to repair them all, and this leads to mutations when the cells divide. Eventually, these mutations lead to cancer. However, UV light is used for disinfecting things like air or transparent fluids or surface objects. The damage to the nucleic acids kills microbial cells. You might have noticed that UV is being introduced into a lot of areas. If you start to pay attention, you'll find UV lights mounted in a lot of restaurants, fast food settings, hospital settings. When you see light sconces that are against the wall pointing upwards and they have a blue glow, those are UV lights. They're there to disinfect the air. The air is flowing around, and any microbes that are floating in the air that come in contact with the light will be subjected to these pyrimidine dimers and hopefully die. It's a way of disinfecting the air. You may start noticing them if you look around carefully. We're about to leave the physical methods and go into chemical methods, so it seems like a good place to stop and transition. We'll pick up with part 3 and the chemical methods of microbial control.